This talk is intended for medical students and residents starting their neurology rotation. It is about anatomy and physiology of the nerve striated muscle junction. The purpose of this talk is to explain how this junction works in order to later understand the pathological conditions affecting it. This talk will be conducted in a question and answer format. The first question is, a motor unit is defined as a neuron, its axon, the axonal branches, and all striated muscle fibers they innervate. A true, B false. The functional anatomy of striated muscle, based on Cherryton's model published in 1906, has a simple arrangement. An upper motor neuron in the cerebral cortex, labeled in this frame 1, makes contact with a lower motor neuron in the brainstem or spinal cord, labeled in this frame 2. The lower motor neuron makes contact with striated muscles. This arrangement allows us to understand a great deal of striated muscle physiology and pathophysiology. Striated muscles are present in the somatic effector system. The somatic effector system is present in cranial nerves and spinal nerves. Striated muscles are also present in branchial system. The branchial system is only present in cranial nerves. So now let's look at an striated muscle and its innervation. This drawing represents an striated muscle. Here we have cut it in half and turn one half towards us to see its inside constitution. The most important constituent of muscle is muscle fiber. Muscle fibers go from one end to the other end of the muscle most of the time. The structure I have just introduced corresponds to a lower motor neuron, or the second neuron in Cherryton model, as you can see in this frame. Now I am pointing to the axon and now to the contact between the axon and the muscle, as it was represented in the initial Cherryton model, but the reality is a bit different. The difference is that the axon goes inside the muscle, and there the axon breaks into multiple branches. These branches innervate many muscle fibers far apart from each other, as represented in this frame. So, by definition, the term motor unit is used for all muscle fibers innervated by one axon, the axon, and the neuron from which the axon originates. So the answer to this question is A. Next question. Muscle fibers in adults are innervated by only one endplate, except in rare occasions. A true, B false. The neuromuscular junction of all striated muscle, somatic or branchial, are constructed the same way. In this frame, we can see a photograph of one axon breaking down into multiple branches. You can see that each muscle fiber receives a single nerve terminal. This one-to-one relation between nerve terminal and muscle fiber is the rule in the adult. only in rare occasions, such as it is in the case of the sartorius muscle, one muscle fiber may receive two nerve terminals. So the answer to this question is A. Next question. The neural component of the end plate is devoid of myelin and Schwann cell coverage. A true, B false. This is the same figure we saw before. Here I have enlarged the photograph, and here I have schematized it. Now I want you to pay attention to the area I have enclosed inside the brown square, which I have now enlarged. In it you can see a blue structure which represents myelin. The covering represents the Schwann cell with its nucleus. As you can see, indicated by the arrow, the terminal axon close to the muscle, even though the void of myelin is surrounded by a Schwann cell. This is a photograph of an electromicroscopic field of an striated muscle junction. In it you can see the terminal axon. This is a myelinated segment of it. Myelin looks like dark lines, as you can see. Here you see the same axon, also myelinated. And here the segment of the terminal axon is not myelinated. So as you can see, as the axon approaches the muscle fiber, it is going to innervate. The axon loses its myelin coverage, but it does not lose the Schwann cell. The area traced in this frame corresponds to one or more than one Schwann cells. The area shaded here corresponds to the nerve or presynaptic region of the neuromuscular junction. The area colored blue represents the junction or synaptic region, which is about 20 to 30 nanometers wide. And the area colored cream corresponds to the postsynaptic region. This region is rich in acetylcholine receptor channels and voltage-gated sodium channels. The postsynaptic region is a modified segment of the striated muscle. So the answer to this question is B. Next question. The term nerve muscle synapse implies the point of contact of a synaptic button with its underlying muscle plate segment. A true, B false. This is the familiar figure I have been showing you. I have added an aqua rectangle and schematized and enlarged its content as I did before. The purpose of doing so, again, is to continue labeling a few more structures and maintaining some perspective on the relation between these structures. In this frame, the arrow points to a relatively large structure. If you have a good imagination, you would be able to see in the contour of this structure a flower bouquet. The thin axon represents the stems and the wider part the roses. The term flower bouquet is often used to describe the nerve terminal, in addition to the more commonly used term endplate. The term endplate was originally used to refer to the muscle under the nerve terminal, but now it is used for both components together, the nerve terminal and the muscle under it. Here I am providing a window to the distalmost region of the terminal nerve. The structure indicated is a synaptic bulb or button in the bouquet. Now I am pointing to the muscle plate. Theoretically, the electrical activity is recorded exclusively from the postsynaptic muscle area under a single button due to the flow of ions through the acetylcholine receptor channel would be called a postsynaptic muscle button potential. But such potentials can only be obtained on very stringent lab conditions and are not useful. So this term is not in use. I will dwell on this point a little more because I think it is key to understanding the workings of the neuromuscular junction. These drawings are from Koffler and Nichols' book, From Neuro to Brain, a good read for those interested in the subject. The arrow in this frame points to the end plate. The electrical activity that would be picked up by a needle right below the nerve ending intracellularly due to ionic flow through the acetylcholine receptor channel is called an end plate potential. The end plate potential is a graded potential proportional to the amount of acetylcholine that binds to nicotinic acetylcholine receptors in the specialized segment of the muscle below the nerve ending. The new arrow I have just inserted is indicating a single synapse button. In theory, if only a single synaptic button is left in a specimen, that is, if all other synaptic buttons are physically removed from the bouquet but one, upon electrical stimulation of the axon going to it, we would see a graded potential proportional to the amount of acetylcholine released by one synaptic button, that is, by the synaptic button that is left. This activity could be labeled synaptic button potential, as we previously mentioned, but also, as we previously mentioned, an experiment in order to obtain this potential is very difficult to perform, and the term is no longer used. I mention it because I feel it helps to stress 2 points about the neuromuscular junction. These 2 points are that one button muscle complex does not make a neuromuscular junction, and that anatomically the nerve muscle fiber synapse is a collection of many synaptic buttons and the corresponding specialized muscle under it. So the answer to this question is B. Next question. The end plate, although anatomically is made of multiple individual synaptic buttons, physiologically behave as a unit. A true, B false. The arrow in this frame is pointing to an end plate. Notice its anatomical configuration. Here in this diagram, which I have shown you before, you can see a drawing of the anatomical configuration of the presynaptic region. Notice how it is made of multiple buttons held together by twigs of axonal branches, as you see pointed by the arrow in this frame and also here. You have also previously seen this cartoon. I am bringing it back to emphasize the loss of myelin and persistence of Schwann cell in the proximity of the muscle fiber, but more importantly to remind us that anatomically the nerve portion of the end plate is a clustering of synaptic buttons. In this new frame, I have removed the Schwann cell covering the distalmost region of the bouquet and cut through the middle of the synaptic buttons to see their internal anatomical structures. I have done this to introduce a new concept. So far we have been talking about anatomical configuration. Now I would like you to think function. The synaptic bouquet works as a unit. Thus, from a functional point of view, we can represent the multiple buttons as a single structure, as I have done in this frame. This single formation represents the functional unit of the neuromuscular junction. We will use this single function structure to explain the process involved in signal transmission from nerve to a striated muscle. Again, this will be done because although anatomically the end plate is made of many synaptic buttons with their underlying patches of special muscular tissue, from a functional point of view, it can be considered a single unit consisting of a single presynaptic structure, a single junctional structure, and a single special muscular patch. So remember, this is the anatomical configuration of the neuromuscular junction. Now you can see its functional configuration. So the answer to this question is A. Next question. The only type of channel on the postsynaptic region of the striated neuromuscular junction are acetylcholine receptor channels. A true, B false. We will start answering this question using the diagram we have just introduced at the end of the last question. I will now enlarge the distal segment to enumerate its different components. So this is the enlarged basic figure that we will use to describe the synaptic structures. This is the endoneurium that continues with the endomysium. The arrows are pointing to the presynaptic region. Now the arrow is pointing to the neurolemma or plasma membrane of the Schwann cell. Now the arrow is pointing to the synaptic terminal. In it we find mitochondria, voltage-gated sodium channels, and voltage-gated potassium channels in the membrane. I have now introduced voltage-gated calcium channels restricted to the segment of membrane facing the muscle. I have also included the representation of choline acetyltransferase, an enzyme needed for the in situ formation of acetylcholine, and the smooth endothelial reticulum. This structure seems to be of significant importance in the control of calcium in the presynaptic region. Calcium, as we will see, plays a distinctive role in the transmission of nerve signals. Now I will remove all components of the presynaptic region that I have just introduced, but for those I am going to use to explain synaptic transmission. And as you can see, I have kept potassium and calcium voltage-gated channels only, but we need to add other components. I have now introduced potassium ions inside the presynaptic region as green circles, and drawn blue lines close to the calcium channels to represent dense active regions. The dense active regions are the docking areas of the synaptic vesicles. Now I have added the synaptic vesicles. as oval structures with blue and red dots join to indicate acetylcholine. Each synaptic vesicle contains one quanta of acetylcholine, corresponding to about 5,000 to 10,000 acetylcholine molecules. We will now look at the synaptic region. The main gap is called the synaptic gutter or primary cleft. The smaller gap that I'm pointing with the arrow is called the junction of fall or secondary cleft. In the primary and secondary clefts, we find a matrix of thin strands of collagen-like fibers, which harbor, among other things, acetylcholinesterase, the enzyme that breaks apart acetylcholine, rendering it inactive, and calcium ions, as well as sodium ions, which I have not represented in this diagram to avoid overcrowding. The arrow is now pointing to the postsynaptic region. This is a segment of modified muscle. In it, you find nicotinic acetylcholine receptor channels, and right at the bottom of the junction folds, you find voltage-gated sodium channels in great abundance. Sodium channels are also present in the rest of the muscle fiber membrane, but in a lower concentration. So the answer to this question is B. Next question. The entry of potassium to the presynaptic region is one of the factors determining the amount of calcium entering the presynaptic region. A true, B false. I am now reintroducing the figure of the functional end plate that we have used to answer the last question. I have done this to explain the process of releasing acetylcholine. In this frame, I have represented an advancing electrical field by magenta bursts, symbols. This depolarization wave advances in the terminal either by electrotonic conduction from the electrical field generated by the most distal action potential generating site, or by the sequential opening of sodium voltage channels segments in the presynaptic membrane. One way or another, the electrical field advances, reducing the intracellular negativity in the terminal, as you can see. This occurs at a relative constant speed until the whole terminal membrane potential is raised significantly. The consequence of depolarization of the synaptic terminal membranes are twofold. The most important one is that voltage-gated calcium channels reach opening threshold, and they open. The calcium ions in the cleft and surrounding areas enter into the presynaptic region. The second consequence of the depolarization of the synaptic terminal is that the potassium voltage-gated channels also open, as indicated in this frame, and potassium ions leave the cell, making the inside of the cell more negative, once again, and driving the membrane towards repolarization. The importance of this phenomena is that one of the factors determining how long voltage-gated calcium channels remain open is how soon repolarizations occur, and how soon repolarization occurs is at least in part dictated by how long voltage-gated potassium channels stay open. So, Drugs that shorten the time potassium channels are open increase the amount of calcium that enters the presynaptic region. So, when repolarization has occurred to a certain level, calcium channels close, and then potassium channels close. So the answer to this question is B. Next question. An endplate potential results from positive ions entering the neuromuscular junction postsynaptic region through acetylcholine receptor channels. A true, B false. The high intracellular concentration of calcium in the presynaptic region triggers a set of events leading to the acetylcholine vesicles closest to the dense active region in what is called the immediate available acetylcholine store to cluster around the dense active regions, as you see in this frame. And then the vesicles open into the cleft, releasing acetylcholine. Soon after acetylcholine is released, the synaptic vesicle closes by incorporating its wall to the surface membrane of the presynaptic region. Now I'd like to focus your attention on the postsynaptic region. Especially, I want you to pay attention to acetylcholine receptor channels. I have singled one out to describe what happens in all of them. These receptor channels are nicotinic acetylcholine cation channels. Thus, they allowed all positive ions to move across it, depending on their concentration and electrical gradient. But in physiologic conditions, the ion important for our storyline at this point, and the one we will follow, is sodium ions. As you can see in this frame, the acetylcholine receptor channel is closed. It is closed because as of yet, two acetylcholine units that have crossed the junction have not interacted with the receptors. Now shift your attention to the voltage-gated sodium channels at the bottom of the secondary cleft and in the area surrounding the endplate. The sodium voltage-gated channels, which I have now enlarged in this frame, are also closed. They are closed because the muscle membrane is depolarized beyond their opening threshold. That is, the inside of the membrane has a negative potential, and that potential is more negative than the threshold that will drive the voltage-gated sodium channels to open. Let us say for now that the cell membrane potential at this stage is minus 90 millivolts. Now I want you to shift your attention back to the acetylcholine molecules, as indicated by the arrow, which are now interacting with the nicotinic acetylcholine receptor channel. Thus, acetylcholine channel opens, allowing positive charges ions, mainly sodium, into the end plate region. The degree of depolarization of this region brought about by the entry of positive charges will depend on and be proportional to the amount of positive charges going in. The potential so created is called an end plate muscle potential. The end plate muscle potential, or just called the end plate potential, does depend on the number of acetylcholine receptors open and the time they remain open, which in order to work normally has to be just right. Closing too fast is bad, closing too slow is also bad. In normal situations, the entry of positive charges will raise the membrane resting potential, from minus 90 millivolts to about minus 40 millivolts. So the answer to this question is true. Next question. The depth of the secondary junction is for the muscle fiber as the initial segment is for the neuron. A true, B false. This question compares the neuromuscular junction to a neuron. Specifically, the bottom of the secondary cleft region to the initial segment of the neuron. Let us first look at the initial segment of the neuron. I hope you recall that neurons receive multiple synapses, some inhibitory and some excitatory. But if at one instant in time the totality of the signal received by the neuron is enough to raise the membrane potential at the initial segment, the richest area in voltage-gated sodium channels of the whole neuron, to less than the threshold of the voltage-gated sodium channels present in this area, an action potential will occur that will travel under physiologic conditions to the end of the axon. I will walk you through this process in the next few frames. I have now removed all the synaptic connections so we can see the body of the neuron. The neuron here represented is a central nervous system myelinated neuron. The area of birth of the axon indicated by the arrow is called the axon hillock. In it, you find a region with a very high concentration of voltage-gated channels called the initial segment. It is there that the initial high voltage electrical field is generated as the result of the simultaneous opening of the many voltage-gated sodium channels present in the region, leading to the initial action potential that in myelinated fibers advances from the initial segment to the first notoranvier and then to the next one, in response to the electrotonic movement of charges within the axon. Or in the case of unmyelinated neurons, go from the initial segment to the adjacent segment in the axon in response again to the electrotonic movement of charges within the axon. Now that we have looked at the initial segment of the neuron, I want us to look at the neuromuscular junction, especially at the sodium channels at the bottom of the secondary clefts. As you recall, we left the storyline of neuromuscular transmission at the opening of the acetylcholine voltage-gated channels. The entry of positive charges and the membrane potential going from minus 90 to minus 40 millivolts, followed by the closure of the acetylcholine channels. Now let's look at the voltage-gated sodium channels voltage sensor. When the voltage-gated sodium channel threshold for opening is reached, which is usually in the range of 15 millivolts above the resting membrane potential, the gates of the voltage-gated sodium channels that so far have been closed open. The reason voltage-gated sodium channels are at the bottom of the junction cleft open before any voltage-gated channels in the rest of the muscle fiber is because of their location. They are located in the proximity of the acetylcholine receptor channels, and also because of the high concentration of voltage-gated sodium channels in this area. When these channels open, sodium comes into the postsynaptic region, producing a high-intensity electrical field that I am depicting by the magenta burst symbols and will be recorded as an action potential. That in physiologic condition will spread to the neighboring areas of the muscle and travel from there to each end of the fibers at the speed of 3 to 5 meters per second. The recording of the initial high voltage electrical field under the end plate and the recording of the electrical field as it travels in the muscle fibers are called muscle fiber action potentials. So the answer to this question is true, A. Next question. One quantum of acetylcholine consists of 5,000 to 10,000 molecules. A, true, B, false. Although I have so far represented the acetylcholine vesicle with 2 molecules of acetylcholine, the amount of acetylcholine in a vesicle ranges from 5,000 to 10,000. This amount is considered a quantum. So the answer to this question is A. Next question. Fetal and adult nicotinic acetylcholine receptors have the same subunits. A, true, B, false. So far, we have represented the acetylcholine receptors as having 5 equal elements. But it is not so. Fetal acetylcholine receptor channels have 2 alpha, 1 beta, 1 delta, 1 gamma subunit. Adult acetylcholine receptor channels have 2 alpha, 1 beta, 1 delta, 1 epsilon subunits. The receptors are in the alpha subunit close to the epsilon and the delta subunit. So the answer to this question is B. Next question. Muscle-specific kinase protein is part of the acetylcholine receptor channel anchoring system. A, true, B, false. Acetylcholine receptor channels in the postsynaptic region need to stay in direct line to the sites of acetylcholine release to be effective. To do so, the acetylcholine receptor channel has to be anchored to the muscle fiber and to the junctional matrix. This anchoring system requires a complicated system of proteins. The proteins that accomplish this important task are rhabsin, DOC7, muscle-specific kinase, LRP4, and agrin. So the answer to this question is A. Next question. The duration of calcium voltage-gated channel opening in the presynaptic region is inversely related to the duration of K voltage channel opening in the same region. A, true, B, false. The duration voltage-gated potassium channels are open, represented for a longer time in the upper part of this panel and for a shorter time in the lower part of this panel impacts the opening time of calcium channels. When potassium channels are open for a long time, calcium voltage channels are open for a short time. When the voltage-gated potassium channels open time is short, then the voltage-gated calcium channel open for a relatively longer time. This relation has been explained by saying that the fewer potassium ions leaving the intracellular space, the longer the intracellular positivity will be sustained, and It is the presence of the membrane relative positivity compared to the membrane at rest that keeps the voltage-gated calcium channels open. It is important to remember that the longer voltage-gated calcium channels are open, the more calcium will go inside the presynaptic region. This is depicted in this frame by the smaller number of blue balls inside the cell in the top panel when the calcium voltage-gated channels are only open for a short time, and by the higher number of blue balls in the lower panel when the voltage-gated calcium channels are open for a longer time. The consequence of lower intracellular calcium is limited release of acetylcholine molecules, as you can see in the upper panel. Acetylcholine molecules are represented by the red and blue balls in the junction. The consequence of higher intracellular calcium is the release of many acetylcholine molecules. So the answer to this question is A. Next question. The calcium that enters the presynaptic region following a single depolarization stays in the presynaptic region for 300 milliseconds. A true, B false. As you can see, I have placed 3 terminal synapses in this frame. They represent 3 stages of acetylcholine release. The first figurine corresponds to a synaptic terminal prior to being stimulated. The second after being stimulated. The stimulus being represented by the flaming thunder. Notice the increased number of blue dots. The blue dots represent calcium ions. In the third figurine, acetylcholine has already been released. The line with the numbers indicate time in milliseconds starting when acetylcholine is released. The y-axis represents the percentage of calcium in the presynaptic region. The amount of calcium entering the synaptic terminal after an stimulus will be considered 100% for the purpose of this diagram. In about 1 millisecond or less after the entry of calcium, the presynaptic region will release, let's say, 60 vesicles. 10 milliseconds after calcium enters the presynaptic region, the amount of intraterminal calcium drops by a certain amount. And the decline continues to the point that no significant amount of calcium is present by 100 milliseconds after it was initially released. I am using 100 milliseconds as the time it takes for calcium drop to non-significant intracellular levels, but this process may last up to 200 milliseconds. So the answer to this question is B. Next question. At a stimulus interval of 70 milliseconds, the number of acetylcholine vesicles released will be more than at a stimulus interval of 300 milliseconds. A true, B false. So, in the scenario we saw in the last question, where we to wait for 110 milliseconds from the first stimulus to give a second stimulus, the whole process would start again with similar results. Calcium, the same amount as before, will enter the terminal after the first stimulus of the second set, again reaching 100%. 1 millisecond later, 60 vesicles of acetylcholine would be released the same as before. And the fate of the intracellular calcium will also be the same, a continuous reduction till no significant presence after 100 milliseconds. In this frame, I have left drawings representing the time interval between the first and the second stimulus, as you can see. In this case, it is 110 milliseconds. I have done so to emphasize that in the second experiment, the intervals are much shorter. In the second experiment, the second stimulus is given 70 milliseconds after the first stimulus. After the second stimulus given 70 milliseconds after the first, as we just mentioned, the same amount of calcium that goes in after the first stimulus goes in, but it will be added to the calcium still there from the first stimulus. So now the amount of calcium present would be more. Instead of 100%, it will be 110%. And the number of vesicles released would also increase, let's say to 66. Calcium will then drop in the same proportion as after the first stimulus. So were we to stimulate a third time 70 milliseconds after the second stimulation, the same amount of calcium will come in as with the first and second stimuli, but the total intracellular calcium will be increased. So will be the number of vesicles released. So the answer to this question is true. Next question. All acetylcholine vesicles are available for immediate release after stimulation. A true, B false. As you recall, we already mentioned the term acetylcholine store some minutes ago. That is because acetylcholine in the presynaptic region is kept in stores. There are actually 3 stores. The first one is called the primary store or the immediate available store. It has about 1,000 vesicles of acetylcholine. The immediate available store is located close to the junction, and as the name indicates, it is readily available to be released after a stimulus. The second store is called the secondary store or mobilization store. It consists of approximately 10,000 vesicles, and it is there to resupply the primary store. It takes 1 or 2 seconds for vesicles in the mobilization store to start replenishing the immediate available store when the stimulation is done at a rate of 2 to 3 per second, but much less following faster rate of stimulation. The third acetylcholine store is called the tertiary store or the reserve store, and it has about 300,000 vesicles. This store is in the axon and the cell body. The tertiary store replenish the mobilization store. So the answer to this question is B. The percentage of vesicles released from the immediate available store after each successful axonal stimulation is about percent. A 10, B 20, C 30, D 40. One score, 20. So the answer to this question is B. Next question. The safety margin of neuromuscular transmission is a function of the difference between the depolarization caused by the opening of acetylcholine receptor channels, normally at the crest of the junction folds, and the depolarization required to activate the voltage-gated sodium channels deployed at the depth of the junction folds. A true, B false. Voltage-gated sodium channels, as we have seen, have sensors. These sensors signal changes in the memory potential to the channel portion of the unit. The channels open when a certain voltage change occurs. In the case of voltage-gated sodium channels that populate the depth of the junctional fold, when the memory potential shifts about 15 millivolts towards zero, that is, when the membrane moves towards depolarization by 15 millivolts, let's say when at rest the membrane potential is 90 and a change occurs that raises the voltage to minus 75 millivolts, gated sodium receptor channels open. A good safety margin is present when the potential generated by the opening of acetylcholine receptor channels drives the membrane potential to minus 40 millivolts. Since the membrane potential has been driven 35 millivolts above the 15 millivolt needed to trigger the opening of voltage-gated sodium channels. A bad safety margin is present when the voltage increase barely surpasses threshold, which is the minimum voltage that will trigger the opening of voltage-gated sodium channels. In this example, going from minus 90 to minus 70. The increase in voltage is 20 millivolts, that is only 5 millivolts above threshold. So the answer to this question is A. Next question. A voluntary contraction achieves maximal force when the frequency firing of the individual axon reaches 30 to 40 discharges per second. A true, B false. At maximal contraction, axonal discharges occurred at 30 to 40 times per second. So the answer to this question is true, A. Next question. In a normal person, the safety factor can be made to fail with continuous maximal muscle contraction for 10 seconds. A true, B false. Axonal stimulation an interval of 25 to 33 milliseconds for 10 seconds, which corresponds to a discharge frequency of 40 to 30 times per second for 10 seconds, leads to calcium accumulation and the transfer of acetylcholine vesicles from the mobilizable store to the immediate available store. This transfer, that with a slow stimulation rate of 2 to 3 stimuli per second, starts 1 second after the first stimulus, in the case of rapid stimulation, probably occurs after 4 or 5 discharges, and it is followed after the 10 seconds of contraction by an elevation in calcium content. The phenomenon just described, the accumulation of calcium and priming of acetylcholine reserves, is called post-exercise facilitation. Post-exercise facilitation lasts for 1 to 2 minutes after 10 seconds of exercise. This phenomenon does not alter the result of repetitive nerve stimulation testing in normal adults. Post-exercise exhaustion will occur 2 minutes after 10 seconds of maximal exercise. At this time, though, through a not well understood process, the release of acetylcholine following subsequent stimulation falls. But the fall is not enough not to be overcome by the safety factor in normal individuals. So the answer to this question is B. Next question. Under physiologic condition, it takes dash milliseconds or less from the time acetylcholine is released into the junction till it is deactivated. A3, B6, C9, D10. We will approach this question from two perspectives. The broad perspective, looking at the neuromuscular junction as a whole, will be addressed first. You can see in this representation the presynaptic region with its cell membrane, the synaptic region with its junction matrix, and the postsynaptic region. I have now added yellow curvy lines to represent acetylcholinesterase at the junction. And I have also added acetylcholine. Now acetylcholine comes in contact with acetylcholinesterase. And acetylcholine is deactivated and converted into acetate and choline. From the time the bulk acetylcholine is released to the time it is broken down, it is about 2 to 3 milliseconds or less, depending on the species and the muscle studied. The second perspective that I like to show you is that of the actual acetylcholine receptor channel. Here we have represented acetylcholinesterase, acetylcholine, acetylcholine receptor channels, and time. In this frame, the acetylcholine receptor channel is closed. The time lapse after the release of a lucky acetylcholine molecule to reach the presynaptic region, activate the receptor, dislodge from the receptor, and be broken down by acetylcholinesterase is about 2 milliseconds. I use the word lucky because a great number of acetylcholine molecules are broken down by acetylcholinesterase shortly after release, and they do never get to reach the receptor. Let's look at the timing of this event more closely. It usually takes for the lucky acetylcholine molecule about half a millisecond to reach the receptors and interact with it. Then the gates of the acetylcholine receptor opens, and for about 1 to 1/2 of a millisecond or less, the gate remains open. Then acetylcholine gets dislodged from the acetylcholine receptor channel by an intrinsic mechanism of the channel, and the gate closes. Acetylcholine is then captured by acetylcholinesterase and broken into acetate and choline. Acetylcholinesterase is capable of breaking up to 5,000 acetylcholine molecules per second. So the answer to this question is A. Next question. After acetylcholine is broken down by acetylcholinesterase, acetate is retaken by the presynaptic region. A true, B false. Early on in this talk, when we listed the presynaptic region components, we mentioned choline acetyltransferase. Choline acetyltransferase is an important enzyme. It produces acetylcholine at the presynaptic region in order to fill the vesicles that would later be discharged, which I have represented here as being empty. In the presynaptic region, in addition to choline acetyltransferase and vesicles, we have acetyl-CoA, here represented by red circles. And now I have represented acetate and choline at the junction. Now I have introduced a cartoonish representation of choline transporter unit that brings the choline from the junction into the presynaptic region. Once inside the presynaptic region, choline travels towards the choline acetyltransferase and is induced by the enzyme to join acetyl-CoA, thus creating an acetylcholine molecule. Then the newly formed acetylcholine molecule, using a vesicle acetylcholine transporter, enters the vesicle. So the answer to this question is false. Next question. Acetylcholine release only occurs with terminal axon depolarization. A true, B false. There are three types of acetylcholine release from presynaptic region. Two of them are quantal release and one is non-quantal. One type of quantal release occurs following synaptic terminal depolarization. Here I have represented synaptic terminal depolarization, followed by calcium entry, and 1 millisecond later by the opening of vesicles into the cleft, and the release of about 60 vesicles of acetylcholine. Each vesicle contains 1 quanta. The second type of release is not related to depolarization. It occurs at a frequency of about 1 every 5 seconds or 1 every second, depending on the muscle studied. Here I have represented a terminal potential at rest. Then, at a rate of 1 every second or 1 every 5 seconds, depending on the muscle, as we have just mentioned, one vesicle opens to the cleft, releasing 1 quanta of acetylcholine, which produces a postsynaptic depolarization of 1 millivolt when intracellularly recorded. This potential is called a miniature endplate potential, usually labeled MEPP. The third mode of acetylcholine release is called non-quantal because it does not go out in packages or in vesicles as all the others. Here I have represented the end of a single quanta release, but it can also occur following post-depolarization release. Following either of these events, while the wall of the vessel is being incorporated into the presynaptic membrane, the vesicle transporter is still pumping acetylcholine through the vessel's wall. But since the vessel is open to the cleft, the newly arrived acetylcholine, instead of staying in the vesicle, float into the cleft. So in this case, an individual acetylcholine molecule or a few of them go into the cleft. So the answer to this question is B. Next question. A compound motor action potential to be clinically relevant must be recorded following supramaximal nerve electrical stimulation. A true, B false. This specimen corresponds to a nerve entering a muscle, breaking into axons, at least 2, then branching into many terminal axons, each forming a bouquet that innervates a single muscle fiber. The new figure figure I have just introduced corresponds to a terminal branch leading to a bouquet and its muscle fiber. And the third element corresponds to an axon twig, which, as you recall, is the unmyelinated segment of axon that ends in or often connects the individual buttons. Also, in this figure, you can see 3 buttons. Hopefully, the illustration by the side, taken from a prior figure I showed you, will help you see what I'm talking about. I have now introduced a muscle cut so we can see the muscle fibers of variable diameters. The color of the borders of the fibers matches the axons constituting the nerve, as you can see. I have now introduced a novel element to represent the end plate zone, that is the zone where you find the buttons from many bouquets, the bouquets from many terminal axon branches, the terminal axon branches of many axons that constitute the nerve innervating the muscle. The pink line represents the skin. I have now added a machine to record electrical activity. Now the muscle electrical activity is being recorded by the needle inside the muscle away from the end plate zone. The electrode inside the muscle is being referred to a surface electrode placed over the tendon at the muscle being studied. When no effort is being made by the patient to move a normal muscle, the needle is not being moved either. The machine does not record any activity nor it makes any sounds. If the patient makes minimal effort to move the muscle, a single motor unit action potential appears. As effort is intensified, a second motor unit action potential appears. Notice that the color represented the motor unit generating it, and that the letters are bigger, representing the recruitment of bigger muscle fibers as the level of contraction increases. And when effort intensifies, a third motor unit appears, and so on. If instead of putting the needle in the muscle away from the end plate zone, the needle is inserted in the end plate zone of the muscle, the patient will complain of severe pain and we will hear a noise and see wiggly lines appearing in the machine. The noise and the lines have been linked to extracellular recorded electrical activity produced by the release of single vesicles in the many end plates around the needle. The single vesicles release are occurred at about 1 every second or 1 every 5 seconds. This activity represents end plate noise. If, as the needle was introduced in the end plate zone, a nerve fiber was irritated by the tip of the needle, then the release of acetylcholine from multiple vesicles produced by the opening of the voltage-gated channel will create an end plate spike. The end plate spike is not the same as an end plate potential. The end plate spike is an all or none phenomenon with invariable characteristics recorded extracellularly. The end plate potential is a graded response corresponding to the amount of acetylcholine release recorded intracellularly. If now we conduct a new experiment, this time not recording from the muscle extracellular region, but with a very thin needle introduced in a muscle fiber just under the neuromuscular plate, and we stimulate the axon terminal going to it. At the very beginning, we see a rather slow rated raise due to end plate potential leading to a muscle fiber action potential. If the needle is kept at the same place but the voltage-gated sodium channels are blocked, then we will only see the end plate potential. You will hear more about this in a few minutes. If the needle is kept in the same place but the nerve is not stimulated, we will record very small potentials called miniature end plate potentials, which is believed to correspond to the non-depolarizing quantal release. So far, we have spoken of experiments requiring the use of needles. The next test does not require a needle. It is done using surface recordings. This is the preferred method to study nerve and muscle, especially in children. This method is used to obtain the recording of the electrical activity traveling through the muscle. It is called a compound muscle action potential. A compound muscle action potential obtained for clinical use must be recorded using a stimulus 20% more intense than the one needed to involve all the fibers in the muscle, as judged by the amplitude of the potential recorded in the muscle. So the answer to this question is true. Next question. Results from muscle fiber recording following nerve stimulation made from the end plate and from 2 centimeters distal to the end plate look the same. A true, B false. These are recordings from a paper published by Fatt and Katz in 1952. The recording from the end plate of the muscle fiber shows miniature end plate potentials. Recording from the muscle fiber away from the end plate shows no potentials. Upon a stimulation of the nerve and recording from the end plate, we will see a potential with a hump preceding the fast rise. The hump is due to the flow of positive ions going through acetylcholine receptor channels. When the recording is from the muscle fiber away from the end plate, no such hump will be present. This is so because in this location, the triggering mechanism of the action potential is a change in capacitance. So the answer to this question is B. Next question. Tetrodotoxin blocks sodium voltage-gated channels. A true, B false. No talk about neuromuscular junction is complete without mentioning neuromuscular block. Neuromuscular transmission can be interrupted by many drugs and toxins. These drugs and toxins can act at the presynaptic, synaptic, and postsynaptic levels. At the presynaptic region, acetylcholine formation may be interrupted by blocking the reuptake of choline. Choline reuptake can be blocked by hemicholine. Also, in the presynaptic region, blocking can be produced by racking release. Racking release of acetylcholine vesicles occurs with botulism. Also, at the presynaptic level, we may have a calcium entry block producing a neuromuscular block. These occur more often in association to verapamil, magnesium, and lead. The last presynaptic situation that I will mention that may produce conduction block is altered repolarization. Altered repolarization can occur due to inhibition of potassium channels, as can be seen with dendrotoxin and with tetrathilammonium, also known as TEA. At the synaptic region, blocking can occur due to decreased acetylcholinesterase function, leading to excessive acetylcholine producing sustained depolarization. This is often seen with organophosphate poisoning. At the postsynaptic region, acetylcholine receptor channel can be affected by toxins that produce sustained depolarization. Such agents include succinylcholine and decamethonium. In addition to sustained depolarization, acetylcholine receptor channel can be affected by inability to depolarize. This situation occurs with curare and alpha-bungarotoxin. Also in the postsynaptic region, neuromuscular blocking can occur due to voltage-gated sodium channel alteration, making depolarization impossible, as it occurs with tetrodotoxin, saxitoxin, and conotoxin. Conotoxin can also act at many levels of the neuromuscular junction, not only at the postsynaptic level in the sodium channels. So the answer to this question is A. Next question. A smooth muscle postsynaptic region is similar to a striated muscle postsynaptic region. A true, B false. Some salient features of a striated muscle neuromuscular junction are the following. 1. Presynaptic region ends in a bouquet of synaptic buttons covered by Schwann cell. 2. The only neurotransmitter released is acetylcholine. And 3. Postsynaptic region is highly specialized. In a smooth muscle, whether multi-unit type with fewer or no connexons, or single unit with connexons, the presynaptic region is characterized by a string of swelling in the nerve called varicocytes covered by Schwann cells except at the releasing zone of the varicocyte. The neurotransmitter release is either catecholamine or acetylcholine with or without associated substances. The smooth muscle has a uninucleated spindle-like configuration and do not have a specialized postsynaptic region. The acetylcholine receptors in these muscles are muscarinic. So the answer to this question is B. Thank you very much for your attention.